Yes. So very good evening, my dear students. So today I am going to start with the last part of the respiration in plants, and that is about. My dear students. So today I am going to start with the last part of the respiration, and that is about. So today I am going to start with the last part of. There is a technical error. Just wait, let us fix this problem. Okay. So let us start with this topic, which is called as electron transport system. Last day I think we have talked about that. I think it's over. Electron transport system is over. So we are left up with only which part? We are left up with amphibolic pathway, respiratory pathway as amphibolic pathway. There's the last part, and respiratory quotient I think we are left up with this part, তাই তো? এই পার্টটা আমাদের বাকি রয়েছে. So before we start with that, let us, as because this is the last part of the respiratory system, let us talk about respiration in a nutshell. Respiration in plants in a nutshell, and then we are going to get into the last part. Okay? So today we are going to talk about respiratory pathway and amphibolic pathway first. Second, anaerobic respiration. Third, respiratory quotient. So these are the topics that we need to discuss today. Okay? Okay. Chalo. So respiration in plants. What is the meaning of the term respiration? Normally we know that the respiration is a catabolic process. Now what is catabolic process? Catabolic process क्या है? That means it is a destructive process. It's a destructive process where the glucose when gets broken down, in presence of oxygen, it gives rise to some energy. What is the energy that we get normally? It is 38 ATP. So what is the chemical equation? The chemical equation is C6H12O6 plus 6 molecules of O2 will fetch you what? 6 carbon dioxide plus 6 molecules of water plus 38 ATP molecules, which is also equal to 686 kilocalorie of energy. That means it is by the breaking down glucose, हमें क्या मिलेगा? Carbon dioxide हम release करेंगे. At the same time, what we will be getting is, we will be getting 686 kilocalorie of energy, which is very much needed for our body's metabolism. तार माने, think about it. Normally we know that the metabolisms are of two types. One is anabolism and one is catabolism. So what is anabolism? Anabolism is a destructive process, and what is catabolism? Anabolism is a constructive process, and catabolism is a destructive process. So as because by breaking down glucose we are getting energy, we are calling it as catabolic process. So what are the 3 steps which are required for getting this 38 ATP? वो 3 step क्या है जिससे हमें क्या मिलेगा? हमें ATP मिलेगा. The first step is what? Is glycolysis. The second step is the link reaction, and the third one is Krebs cycle, and the third one is Krebs cycle. Now we know it is by the process of glycolysis. So glycolysis का और एक नाम क्या है? It is also called as EMP pathway. And it has come from the 3 terms: Emden, Meyerhof, and Parner's pathway. And the net gain in this reaction is what? 8 ATP. The net gain is 8 ATP. Now I'm not differentiating these 2 terms into substrate level phosphorylation क्या है and oxidative phosphorylation क्या है. हम ये बात नहीं कर रहे हैं. We are not going to talk about that right now. But what is the total number of ATP? The net gain of ATP that we get out of glycolysis is 8 ATP. Now under link reaction we get 6 ATP. And when we talk about Krebs cycle, we get 12 ATP in one Krebs cycle, into 2 will fetch us 24 ATP. So these three together makes 38 ATP molecules. Now, let us get into these details. In glycolysis, अगर आपको याद रहे कितने सारे NAD से NADH2 बना या NADPH2 बना. So NAD has been reduced into NADPH2. This is very very important. Mind you this. In glycolysis, कितने सारे ऐसे NADPH2 लगे? 2 NADPH2 have been formed. 2 of the NADPH2 have been formed, which ultimately when gets inside electron transport system, जब ये NADPH2 ETS में जाता है, इलेक्ट्रॉन ट्रांसपोर्ट सिस्टम में जाता है, अल्टीमेटली इट फॉर्म्स, हम जानते हैं कि एक NADPH2 3 ATP देते हैं. So 2 into 3 it is going to give us 6 ATP. It is going to give us 6 ATP. Any issues with this command? So in glycolysis, this NAD is reduced to give rise to NADH2 or NADPH2, which ultimately ETS में जाने से, what is ETS? Normally we know that ETS के लिए क्या चाहिए? Oxidative phosphorylation. We call this as oxidative phosphorylation. Now Varun, tell me, ETS requires what? ETS requires O2. ETS requires what? Oxygen. तो आज हम क्या पढ़ने वाले हैं? एनारोबिक रेस्पिरेशन. माइंड यू दैट. और एनारोबिक रेस्पिरेशन से अगर ग्लूकोज का एनारोबिक रेस्पिरेशन होगा, कितने सारे एटीपी हमें मिलेगा? दैट इज व्हाट वी आर गोइंग टू टॉक अबाउट टुडे. एनी इश्यूज विद दिस? कम ऑन. क्लियर? नाउ सो इन टोटल यहां पे 6 एटीपी, और हम क्या जानते हैं? 4 ATP ग्लाइकोलाइसिस में डायरेक्टली बनता है. Now who is going to tell me इस डायरेक्टली बनने को क्या कहते हैं? What is that process called? It is called as substrate level phosphorylation, SLP. Substrate level phosphorylation में 4 ATP बनते हैं. Phosphorylation में 4 ATP gets formed. So in total 10 ATP is formed, but out of 10, 2 ATP is being consumed. So net gain is 8 ATP. And I think you know वो 2 ATP कहाँ पे यूज किया जाता है. The first 2 process, ग्लूकोज से ग्लूकोज 6 फॉस्फेट, ग्लूकोज 6 फॉस्फेट से फ्रुक्टोज 6 फॉस्फेट, फ्रुक्टोज 6 फॉस्फेट से फ्रुक्टोज 1,6 बाइफॉस्फेट. दे आर द 4 मेन स्टेप्स जहाँ पे 2 ATP यूटिलाइज हो जाता है. एंड यू कैन सी इन योर बुक इन द वेरी फर्स्ट स्टेप, पेज नंबर 156, इफ आई एम नॉट रॉन्ग, अबाउट ग्लाइकोलाइसिस. About glycolysis, the first 4 steps. Clear? So this was about glycolysis. So what is the main thing that I want to tell it to you? कि glycolysis में कितने सारे NAD का reduction होता है to give rise to NADH2? What is that? 2 of the NADPH2 has been formed, and this NAD has been reduced to give rise to NADH2. The second step is what? The second step is link reaction. So in link reaction, glycolysis से ultimately क्या बना? By the process of glycolysis, glucose 6 carbon glucose से, tell me what has been formed? 2 molecules of 3 carbon compound called pyruvic acid being formed. Fine. 2 molecules of pyruvic acid being formed. Now these 2 pyruvic acid, they are going to give rise to, by decarboxylation, carbon dioxide goes off and they form into what? Acetyl coenzyme A, which is a 2 carbon compound. They form what? Acetyl coenzyme A, which is a 2 carbon compound. Any issues with this? Now this step is called link reaction, but during this step what happens? Again another NAD is being reduced to give rise to NADPH2. Is this clear? Over here again another NAD plus or NADP plus binds with 2 hydrogen ion to make what? NADPH2. So 2 of the NADPH2 when gets inside where? ETS, electron transport system में जाएगा तो क्या देगा? 2 into 3 ATP, that is 6 ATP will be given in this step. Link reaction is going to fetch you 6 ATP. कितना मिलेगा आपको? 6 ATP मिलेगा in link reaction. Clear? And then in the last step, that is the Krebs cycle. So Krebs cycle कितने सारे होंगे? 2 Krebs cycle होंगे, because Krebs cycle is going to take place inside the mitochondrial matrix, and this acetyl coenzyme A is going to start with the 2 carbon compound, and we know that the last compound is what? Oxaloacetic acid. So this ultimately is going to form into, this is the cycle which goes on. So 4 plus 2 is going to give rise to citric acid first, citric acid which is a 6 carbon, then isocitric acid, and you know that, बोलो थोड़ा सा हेल्प करो, citric के बाद isocitric, then cis can crazy intelligent current, cis aconitic acid which is a 6 carbon compound. So can crazy intelligent, that is isocitric acid which is again a 6 carbon compound, can crazy intelligent current, that is alpha keto glutaric acid. Tell me how many carbon compound it is having? 5 carbon. So what is going to happen? Decarboxylation occurs over here. So यहाँ से क्या निकल गया? Carbon dioxide निकल गया. So during this process, again another NADP is going to bind with 2H plus to give rise to reduced form called NADPS2. Now from alpha ketogluteric acid, it's going to make what? Can crazy intelligent current solve. What is solve? It is succinyl coenzyme A, which is a 4 carbon compound. So 5 से 4 carbon compound मतलब फिर से क्या बन गया? यहाँ से carbon dioxide निकल गया. So what is going to happen over here? Again another NADP is going to bind with 2H plus to give rise to NADPS2. So again NADP has been reduced to give rise to NADPS2. Any issues with this? Clear? Now after succinyl coenzyme A, it's going to make into succinic acid. Succinic acid. Again succinic acid is a 4 carbon compound, and what happens over here? One of the GDP binds with one IP to make what? GTP, guanosine triphosphate, and guanosine triphosphate is as good as ATP. Succinic acid is going to change into what? Fumaric acid, and fumaric acid is having again 4 carbon compound. It's having 4 carbon compound. What happens from succinic acid to fumaric acid? Again, what happens? One of the FAD is being reduced to give rise to FADS2. Clear? And normally we know it's again FADS2 is also a form of energy. When it gets inside ETS, it gives you how many ATP? 2 ATP. It gives you 2 ATP. From fumaric acid, it is going to form into what? From fumaric acid, it is going to form into malic acid. And from malic acid, it is going to give rise to oxaloacetic acid. From oxaloacetic acid, ultimately forms into acetyl-CoA. From malic acid to oxaloacetic acid, again one of the NADP is going to bind with 2H plus to give rise to NADPS2. So in total, Krebs cycle में कितने सारे NADPS2 बने? 3 NADPS2 बने. There are 3 NADPS2 being formed, is equal to 3 into 3 ATP is equal to 9 ATP. ETS में जाने के बाद, when it gets inside ETS, it's going to give rise to 9 ATP. Called what kind of phosphorylation? Oxidative or substrate level? Oxidative phosphorylation. Clear? Oxidative phosphorylation. And then one FADS2. So one FADS2 is going to give rise to one FADS2 when it gets inside electron transport system. कितना ATP मिलेगा आपको? 2 ATP. और एक GTP is equal to 1 ATP. So in total you will be getting 12 ATP from one Krebs cycle. ऐसे कितने सारे Krebs cycle होंगे और एक acetyl-CoA बैठा हुआ है, वो भी तो जाएगा Krebs cycle करने के लिए. So that is also going to give rise to 12 ATP. So 12 plus 12, 24 ATP. 12 plus 12, 24 ATP. Now what is most important? 24 plus 6 plus 8 कितना हो गया? 38. ये था balance sheet of breaking down one molecule of glucose. What kind of respiration was that? Aerobic or anaerobic? Aerobic. So right now we are going to talk about anaerobic respiration. किसके बारे में बात करेंगे? Anaerobic respiration. So let us talk about anaerobic respiration first. Anaerobic respiration. So when we talk about anaerobic respiration, we have to start our journey from what? Glucose. कहाँ से start करेंगे? Glucose से start करेंगे. So glucose से क्या बनेगा? Pyruvic acid. ग्लूकोज से क्या बनेगा? Pyruvic acid. कितने कार्बन है? 3 कार्बन है। कितने कार्बन है? 3 कार्बन है। नाउ ये एनारोबिक रेस्पिरेशन कहां पे हो सकता है? ये हो सकता है कहां पे? ये हो सकता है सी फ्रॉम पायरुविक एसिड इफ इट फॉर्म्स इनटू लैक्टिक एसिड, if it forms into lactic acid. How many carbon lactic acid is having? 3 carbon. अब बोलो पायरुविक एसिड को देखने में क्या है? What is the structure of pyruvic acid? It is CH3, C double bond O and C double OH. This is pyruvic acid. और ये क्या है? लैक्टिक एसिड। लैक्टिक एसिड में क्या होगा? यहाँ पे OH हो जाएगा, यहाँ पे क्या हो जाएगा? अल्कोहल ग्रुप आ जाएगा। That means what has occurred over here? Pyruvic acid has been reduced to give rise to lactic acid. But in this step, what has happened over here? NADS2 has been oxidized to give rise to NAD plus. So यहाँ पे क्या हो गया? ऑक्सीडेशन हो गया। यहाँ पे क्या हो गया? ऑक्सीडेशन हो गया। तो एक ही रिएक्शन जहां पे रिडक्शन पायरुविक एसिड से यहाँ पे सेकेंड कार्बन में यहाँ पे हाइड्रोजन ऐड हो जाना, दैट इज गोइंग टू मेक व्हाट? लैक्टिक एसिड। ना वरुण टेल मी, 3 कार्बन से 3 कार्बन डाइऑक्साइड क्या यहां से निकल गया? नहीं निकला। पर यहां पे क्या हो गया? रिडक्शन एंड ऑक्सीडेशन टेकिंग प्लेस साइमल्टेनियसली। टेल मी व्हाट काइंड ऑफ एंजाइम्स हेल्प्स इन दिस काइंड ऑफ रिएक्शन जहां पे ऑक्सीडेशन और रिडक्शन दोनों एक साथ होते हैं? ऑक्सीडो रिडक्टेज एंजाइम, एंड बेसिकली दे आर डन बाय dehydrogenase. So what is the name of the enzyme which is going to help in this? It is lactate dehydrogenase. Lactate dehydrogenase enzyme is going to help in this. अब मुझे बोलो, ये वरुण बोलो, ये जो NAD बना, ये NAD बनने के समय ये NAD क्यों बना? ये NAD बना, देखो ग्लूकोज से पायरुविक एसिड बनने के प्रोसेस में कितने सारे नेट गेन होता है एटीपी का? 2 ATP ना। आप बोलोगे सर आपने तो अभी बोला 8 ATP। वो 8 ATP में हमने किसको हिसाब में रखा था? ETS। वी हैव टॉक्ड अबाउट ETS, है ना? इन केस ऑफ 8 ATP, हाउ मेनी NADPS2 being formed in glycolysis step? 2 NADPS2. So 2 NADPS2 is going to go inside ETS, और ETS ऑक्सीजन के बिना होगा नहीं होगा? नहीं होगा। That means we cannot consider these 2 NADPS2 वो कभी ऑक्सीजन के बिना anaerobic respiration में ATP नहीं बनाएगा। But why we have written 2 ATP? Total कितने सारे ATP बनते हैं directly? Tell me. How many ATP being formed directly? Durva, are you getting me? How many ATP being formed directly in glycolysis step? Substrate level phosphorylation. How many ATP? And how many of ATP you are going to deduct from here? 2 being consumed. So 4 minus 2. 2 ATP is the net gain. 2 ATP is the net gain. So over here 2 ATP is the net gain. For glycolysis to occur, what it needs? NAD plus. In case of anaerobic respiration, lactic acid forms where? Inside muscles. Lactic acid forms by anaerobic respiration. It forms where? Inside muscles and in some bacteria, and in some bacteria is prokaryote. This anaerobic respiration can take place. So by anaerobic respiration, how many ATP is the net gain? 2 ATP. Because in anaerobic respiration, what is never possible? ETS is never possible, because ETS के लिए इलेक्ट्रॉन ट्रांसपोर्ट के लिए what is required? Oxygen. And we are talking about what? Anaerobic respiration. So this is the process. Is this clear? This takes place where? In muscles and in bacteria. But when we talk about yeast, from glucose, which is a 6 carbon compound, it forms into what? It forms into pyruvic acid, which is a 3 carbon compound. Now think about it. Ultimately, how many ATP being formed over here? 2 ATP being formed. 2 ATP being formed. Now from pyruvic acid, if this takes place in yeast cell, this takes place in yeast cell. So from pyruvic acid, that is CH3, C double bond O, C double OH. From this pyruvic acid, it first forms into acetaldehyde, CHO. It forms into what? Acetaldehyde. Now you tell me, Varun, how many carbon this pyruvic acid is having? How many carbon acetaldehyde is having? 2 carbon. So what has happened from here? What has gone out from here? Carbon dioxide. So decarboxylation has occurred. What has happened? Decarboxylation. What is the name of the enzyme which is going to help in this? Pyruvate decarboxylase. Pyruvate decarboxylase is going to change pyruvic acid into acetaldehyde. And this acetaldehyde is going to ultimately form into what? Into ethanol. That is CH3, CH2, OH. This is ethanol. क्या बन गया? Ethanol बन गया. What has been formed? Ethanol has been formed. And mind you, जब ये पायरुविक एसिड से ये एसिटाल्डिहाइड बना तो क्या होगा यहाँ पे? Pyruvate decarboxylase has been formed. And again, in this reaction, ultimately क्या बना? Ethanol बना. But during this step, what is going to take place? Again, NADH2 is going to oxidize into, sorry, not in this step, not in this step. So decarboxylation occurs over here. So what is going to happen over here? In this step, one of the NADH2, NADPH2 is going to form into NAD+. So what has happened over here? Oxidation. And what is this reaction? This reaction is called reduction from acetaldehyde to ethanol. Hydrogen being added called reduction. During this step, क्या होगा? बोलो. What is going to happen? NAD is going to make NAD+. This NAD+ किस लिए बनाया है? क्योंकि glycolysis नहीं होगा without NAD+. Durba, tell me why you are not going to get glycolysis without NAD+. Because in glycolysis, NAD is going to bind with 2H+ to make what? NADPH2. कितने सारे ऐसे होंगे? 2 NAD bind करेंगे to make 2 NADPH2. So to make this continuation, that is ये anaerobic respiration glycolysis step को continue करने के लिए होता है. This anaerobic respiration helps in maintaining glycolysis. It helps in maintaining glycolysis because without NAD+, you cannot have a complete glycolysis step. Is this clear? Are you getting me? Arjit, any issues with this? So ball, what has happened over here? What is the net gain again? In anaerobic respiration. And this ethanol which is produced, ये ethanol के साथ क्या निकल गया? Carbon dioxide निकल गया. This ethanol which is produced, it takes place where? In which cell? Yeast and in some of the bacteria. In some of the bacteria, the same process takes place. This is called anaerobic respiration. This is called anaerobic respiration. Can you tell me what is the name of the enzyme going to be? यहाँ पे क्या हो रहा है? Reduction, oxidation और reduction दोनों हो रहे हैं. Durba, tell me what kind of enzyme is going to take place? What kind of enzyme is going to do this function? Oxidoreductase. And oxidoreductase is done by always which enzyme? Dehydrogenase. Which enzyme? Dehydrogenase. So the name of the enzyme is over here. The name of the enzyme is alcohol dehydrogenase. The name of the enzyme is alcohol dehydrogenase. Is this clear? Any issues with this? Come on. বুঝলাম না বাবা হয়েছে কোনো অসুবিধা হয়েছে? Yes, any issues with this? So let's again wrap it up. So what was the condition? The condition was ki porchilam amra, we were talking about respiration in plants. It's a catabolic process. It's a destructive process. Glucose gets broken down in presence of oxygen to give rise to 38 ATP. So it's an aerobic respiration. So this is the reaction. Glucose in presence of oxygen is going to give rise to 6 molecules of carbon dioxide plus 6 molecules of water. Along with that 38 ATP is going to come out, which is equal to 686 kilocalorie of energy. The next step is what? Glycolysis, which is also called as EMP pathway. Total net gain is 8 ATP. In link reaction, there is a 6 ATP being formed, and by Krebs cycle 12 into 2, that is 24 ATP. So in total, this is the balance sheet how one molecule of glucose can give rise to 38 ATP molecules. Any issues with this? Any problem? Come on. Yes? Great. And then next what we have studied? Next we have studied that in glycolysis, NAD is reduced. In glycolysis, NAD is reduced. See, NAD is reduced. In glycolysis, NAD is reduced. Okay? So NAD is reduced. So this is NAD is reduced to NADPH2. Fine. 2 NADPH2 is being formed in glycolysis. And when it goes inside ETS, it gives rise to 6 ATP, and 4 ATP we get it directly by substrate level phosphorylation. কিভাবে পাই আমরা? Substrate level directly যদি ATP তৈরি হয়, if we get directly ATP with that process is called as substrate level phosphorylation. Any issues with this? Come on. And then if it goes inside ETS and gives rise to ATP, we call it oxidative phosphorylation. And for ETS, what is the major requirement? The major requirement is what? O2. The major requirement is O2. So 10 minus 2, what is 2? 2 is being consumed, 2 ATP is being consumed. So 10 minus 2 is going to be 8 ATP is the net gain. Link reaction, in link reaction, what happens? The pyruvic acid changes into acetyl-CoA. So this step is called glycolysis. Glucose 2 pyruvic acid is called glycolysis, and this takes place where? Inside the mitochondrial matrix. And link reaction, what happens in this reaction? 3 carbon pyruvic acid gets changed into 2 carbon acetyl-CoA. So what happens over here? 1 carbon dioxide goes off by decarboxylation occurs, and it gives rise to what? Again, NADP is being reduced to give rise to NADPS2. So you can get over here 2 of the NADPS2 over here. Fine. Next, Krebs cycle. So if 2 of the NADPS2 is formed, what is the net gain of ATP? The net gain of ATP, if it goes inside ETS, again for ETS, what is requirement? What is the requirement? O2 or no oxygen? O2 is required. So oxygen is required. So ultimately you'll be getting 6 ATP. And in Krebs cycle, in this reaction, can crazy intelligent current solve some foreign mafia operations that we have already done in the earlier classes? Jodi na dekhe thakis, just go and watch that. And 2 of the Krebs cycle is going to ultimately form into what? 24 ATP. 12 into 2. But for that, majority of ETS has got a role. ETS is going to only make its role if it gets oxygen. Why we are talking about all this? Because we need to understand about what is anaerobic respiration. So in anaerobic respiration, कोनटाके माइनस करे भाबो, किसके बारे में हम सोच ही नहीं पाएंगे? ETS के बारे में हम सोच नहीं पाएंगे. ऑक्सीजन नहीं रहेगा तो ETS नहीं होगा. तो ये इसमें कितना ATP बनता है? एनारोबिक रेस्पिरेशन में 2 ATP बनता है. एंड व्हाट इज द रीजन फॉर दैट? द रीजन इज glucose is going to give rise to pyruvic acid, and for that 2 ATP is going to get formed. 4 minus 2, 4 ATP being formed directly by substrate level phosphorylation, minus 2 ATP which is being consumed. So 2 ATP is the net gain. But this pyruvic acid is going to change into lactic acid. This pyruvic acid is going to get changed into lactic acid compound. So from pyruvic acid, which is a 3 carbon compound, you are getting what? Lactic acid, which is also a 3 carbon compound. So there is no decarboxylation, but pyruvic acid is having CH3COCOH. This CH3COCOH, what happens in lactic acid? In this second carbon, what is present? Alcohol group, OH group. That means it has been reduced. Addition of hydrogen has occurred. So that is called reduction. So pyruvic acid to lactic acid is a reduction process. But in this step, NADS2 is being oxidized to give rise to NAD+. This NAD+ is required in glycolysis so that it can continue the glycolysis step and can make ATP. So how many ATP is the net gain in case of anaerobic respiration? 2 ATP is the net gain if the product is what? Lactic acid. And this is going to take place where? Inside muscles. Inside muscles and inside some of the bacteria called prokaryotes. Another way that glucose is broken down into pyruvic acid. 2 ATP is the net gain, but this pyruvic acid now changes into acetaldehyde. For that, what has happened? Pyruvic acid is having 3 carbon, acetaldehyde is having 2 carbon. So what has happened over here is carbon dioxide has moved off. So called decarboxylation has occurred. And what happens? What is the name of the enzyme? The enzyme is pyruvate decarboxylase. This acetaldehyde gets reduced to give rise to ethanol. But during this step 1, and again another NADPS2 is going to get oxidized to NAD+. It's oxidized to NAD+. Clear? And for this, what is the enzyme required? Alcohol dehydrogenase. Why dehydrogenase? Because oxidation and reduction taking place simultaneously. We call it oxidoreductase enzyme is done by dehydrogenase enzyme. So what is the net gain? The net gain is 2 ATP, and this takes place where? In the yeast and bacteria. Any issues with this? That's all about anaerobic respiration. Now see this step. ETS we have already done in the very last class. We have already covered it. Fine, we have talked about F0, F1 particle around. This is the respiratory pathway. See, amphibolic pathway is our next point. Amphibolic, why it is called amphibolic pathway? What is the meaning of amphibolic pathway? Amphi means what? Both. So you can see in your book, this step. So what is the step? This step we already know. This is carbohydrate. This is carbohydrate. Fine? Yes. See, in this step, our journey starts from carbohydrate, that means glucose. So this is glucose, and from this glucose, see, glucose, it forms glucose 6-phosphate, fructose 1,6 biphosphate. This is dihydroxyacetone phosphate, which is called as DHAP, and this is glyceraldehyde 3-phosphate. That is called triose phosphate or glyceraldehyde 3-phosphate. Ultimately forms into what? Ultimately it forms into pyruvic acid, and this pyruvic acid by link reaction, pyruvic acid is a 3-carbon compound and this acetyl-CoA is a 2-carbon compound. So this step is what? Link reaction. This is a link reaction, and after link reaction, acetyl-CoA gets inside Krebs cycle and ultimately forms into how many ATP? 38 ATP. So this is a straight, first step is glycolysis, this step is called glycolysis, this step is called link reaction, and this step is third step called Krebs cycle. Just now we have finished it off. This is the breakdown of glucose. Now, if you are in a fasting condition, the first in your body, whatever carbohydrate you have got, first it is going to get utilized. Carbohydrate is going to get broken down, and then you are going to get the energy by breaking down fats. So the moment you break down fats, the fats will be broken down into fatty acid and glycerol. Glycerol, we know, if you know the structure, glycerol is having how many carbon? 3 carbon. It is CH3OH, CHOH and CH2OH. This is glycerol and this is fatty acid. This glycerol gets broken down, fatty acid. This fatty acid, it comes straight away to acetyl-CoA. So mind you one thing. What happens? The fatty acid, it comes down, it gets straight away into acetyl-CoA. How many carbon acetyl-CoA is having? 2 carbon. So after acetyl-CoA, it will get into what? Krebs cycle. Mind you, respiratory pathway is called as amphibolic pathway. What is the meaning of amphibolic? That means it is both constructive as well as destructive. It is both constructive and destructive pathway. Understand why we call destructive? We know. What is constructive? Sometimes from acetyl-CoA, it can also form into fatty acid. Acetyl-CoA can also form into fatty acid, and when it forms into fatty acid, again is going to be able to make what? ATP by breaking down. So this step is very very important. This step, acetyl-CoA एक बार बन गया तो acetyl-CoA भी क्या बना सकता है? फैटी एसिड बना सकता है. But mind you, glycerol can ultimately form into pyruvic acid. Glycerol is a 3-carbon compound. Glycerol is a 3-carbon compound. It can form into pyruvic acid. It can straight away form into pyruvic acid. That means 2 of the product by breaking down fats, fatty acid and glycerol are the products. Fatty acid can make directly acetyl-CoA. From acetyl-CoA, it will be getting into what? From acetyl-CoA, it will be getting into Krebs cycle and it's going to make the ATP. And sometimes this acetyl-CoA can again make fatty acid back. And that's why it is called amphibolic pathway. So इसको amphibolic pathway क्यों कहते हैं? What is the meaning of amphi? Both. So पहले destruction हुआ, उसके बाद फिर से construction हुआ. फिर से acetyl-CoA से क्या बना? Fatty acid बना. Now pyruvic acid, glycerol से क्या बन सकता है? Pyruvic acid can get formed. And from pyruvic acid, acetyl-CoA link reaction हुआ. उसके बाद acetyl-CoA से क्या बनेगा? Krebs cycle. So it's going to ultimately form into Krebs cycle. So this is what is going to take place during breakdown of fats. When we talk about protein, last में कौन टूटता है? When you are in a complete fasting condition, first carbohydrate gets broken down, secondly it is a fat which gets broken down, and lastly it is a protein which gets broken down. Now when protein gets broken down, what is the unit of protein? Amino acid. Protein gets broken down into amino acids. These amino acids can straight away form into pyruvic acid. Now if it's a 3-carbon compound, amino acid is a 3-carbon compound, it can directly form into 3-carbon compound called pyruvic acid. Now Durva, tell me, do you think amino acid is always going to have a 3-carbon compound? Sorry, when we break down protein, it is going to have a chain of 3-carbon compound. It can also have a 5-carbon compound. If it is having a 5-carbon compound, then it can make alpha ketoglutarate acid. That means the 5-carbon compound can straight away get into Krebs cycle. Alpha ketoglutarate acid kothay ache? Where it is formed? Where it is formed? Alpha ketoglutarate acid is there inside Krebs cycle. That means this protein, they can ultimately come in this chain of reactions. Either they can straight away form into pyruvic acid, or it can get in straight away into the Krebs cycle with alpha ketoglutarate acid if it's a 5-carbon compound. Well, any issues with this? Got the point? So this is why we call it amphibolic pathway. Why it is called amphibolic pathway? Because from fatty acid it can form into acetyl-CoA, and from acetyl-CoA back we can make what? Fatty acid. Is this clear? It's a very, very important concept, and it's there in your book, you can see over there. The last topic is about respiratory volume. The last topic is about respiratory volume. Respiratory quotient. Respiratory quotient. When we talk about respiratory quotient, what is respiratory quotient is all about? Respiratory quotient means RQ is equal to volume of carbon dioxide evolved by volume of oxygen consumed. By volume of oxygen consumed. Okay, now volume of carbon dioxide evolved by volume of oxygen consumed. When we talk about this, let us talk about this step. Let us talk about this step. Okay, now glucose, when you break down glucose को अगर break down करेंगे in presence of oxygen, you'll be giving rise to 6 molecules of carbon dioxide plus 6 molecules of water plus energy. Now, tell me how many carbon dioxide being evolved? 6. How many carbon dioxide being evolved? 6 by how many oxygen consumed? 6. 6 by 6 is equal to what? 1. So what is the respiratory quotient of glucose? The respiratory quotient, RQ value of glucose is what? 1. So RQ value of glucose is 1. But you can see in some of the fats like tripalmitin, this is tripalmitin. So what you have to remember is the complete formula. C51H98O6. C51, 2 into C51H98O6. How many oxygen consumed? 145 oxygen consumed. How many carbon dioxide evolved? 1 and 2. So what is RQ? What is RQ? 102 carbon dioxide evolved by oxygen consumed is 145 is equal to 0.7. So it is less than 1. So tripalmitin is a fat. Tripalmitin is a fat, and the RQ value, respiratory quotient of tripalmitin is what? 0.7. 0.7 is the RQ value, respiratory quotient of fat. It's very important. In case of protein, it is 0.9. In case of protein, the RQ value is 0.9. 0.9. Now, the lesser the RQ, the more is the oxygen required. The more is the oxygen required. The lesser the RQ, the more is the oxygen required. So break down fat. Lesser is the RQ. That means more is the oxygen required. Is it oxygen consumed? Fine. Got the point? So if the question is, if the RQ value of some agent, some substrate is less, that means it is going to require more oxygen or less oxygen? More oxygen. Any issues with this? That's all about RQ. Let us solve some other questions. Yes. In front of you. This is the first question. Come on. Try. Let's try for the first question. What is that? All enzymes of TCA. What is TCA? Arijit, what is TCA? TCA cycle means what? Tricarboxylic acid cycle. That means Krebs cycle. That means all enzymes of TCA cycle are located in the mitochondrial matrix. True? Except one, which is located in inner mitochondrial membrane in eukaryotes and in the cytosol in all prokaryotes. This enzyme is isocitrate dehydrogenase, malate dehydrogenase, succinate dehydrogenase, lactate dehydrogenase. Lactate dehydrogenase, we have got it where? Just now we have given this information about lactate dehydrogenase. Varun, tell me, कहाँ पे पढ़ा था हमने lactate dehydrogenase के बारे में? Anaerobic respiration. Pyruvic acid gets changed into lactic acid by the help of lactate dehydrogenase. But see, all enzymes of TCA cycle are located in the mitochondrial matrix except one, which is not there in the mitochondrial matrix. It is a succinate dehydrogenase which is not there in the mitochondrial matrix. Succinate dehydrogenase is the enzyme which is not present in the mitochondrial matrix. What is this? The overall goal. The overall goal of glycolysis, Krebs cycle, and the electron transport system is the formation of this one. The overall goal of glycolysis, Krebs cycle, and the electron transport system is the formation of
ATP in small stepwise units.
Yes, ADP in small stepwise unit. ADP is going to get produced, but in a small stepwise unit. What is a small stepwise unit? Glycolysis, Krebs cycle, and link reaction, electron transport system. ये सब क्या है? यहाँ पे क्या होता है? Small stepwise step होता है. Ultimately, what is the ultimate goal? To make what? To make ATP. To make ATP. Next, chemiosmotic theory of ATP. Chemiosmotic theory of ATP synthesis in the chloroplasts and mitochondria is based on, I think this is very easy. Chemiosmotic theory of ATP synthesis in the chloroplast and mitochondria is based on, remember, what is being made? What is being made? Proton gradient is being made. So this is the outer membrane. This is inner membrane. This is inner membrane. So what is being made? What is being made? If this is inner membrane, ETS is present over here. Last class we have already talked about this. Remember, this is complex 1, complex 2, complex 3, and this is complex 4. Fine. If the enzyme, if NADS2 is going to come over here, it's going to give rise to what? NAD plus is going to give rise to electron. And when this electron will be carried by ubiquinone, remember, we have talked about that. Last जो वीडियो हम बनाए थे उसको थोड़ा देख लेना. Last video, that's respiration number 3. We have talked about ETS. This is UQ, ubiquinone. It is going to accept this electron. While accepting, कितना हाइड्रोजन को धक्का देकर पुश करेगा from the matrix towards the inner membrane space? 4 hydrogen. 4 hydrogen is going to be pushed. The proton gradient has been made in the inner membrane space. In the inner membrane space. Matrix থেকে ধাক্কা দিয়ে হাইড্রোজেন কে প্রোটন কে এখানটা একটা পুল তৈরি করা হবে. What has been made over here? A pool has been created. Fine. If the substrate that we have got is NADS2, then this is going to happen. This is this complex 1 made by NADH dehydrogenase. Clear? And when this ubiquinone is going to transfer it over here in complex 2, sorry, ubiquinone is going to give directly to the complex 3, and from complex 3 it is going to give rise to what? 4 hydrogen again going to be get pushed. 4 hydrogen gets pushed over here, and from complex 3 to complex 4. So from complex 4, when the electron comes over here, the final fate of the electron is half O2 plus 2 of the electron plus 2 hydrogen is going to make what? H2O. What is being said about complex 4? What is the process called? Terminal oxidation. What is that process called? Terminal oxidation. So ultimately 4 plus 4 plus, from here 2 hydrogen is being posed. So in total 10 hydrogen being posed by 1 NADPS2 and 1 NADPS2. So, and there is, what is present over here? Complex 5. And what is that? F0, F1 particle. F0, F1 particle. Hydrogen can only come back to the matrix through this channel called F0F1 particle. With every 4 hydrogen ion, one of the ATP is going to get formed. So out of 10 hydrogen, 2.5 ATP, that is equal to 3 ATP is being formed. Due to what? 1 ATPs2. So কি তৈরি করা হয়েছে এখানটা? Proton gradient. So what is the answer going to be? What is the answer going to be? Proton gradient. Check it out if you have got any doubt in the ETS. ETS is very, very important. আমরা আগের রেসপিরেটরি সিস্টেমের রেসপিরেশন ইন প্ল্যান্টসের পার্ট নম্বর 3 তে করে ফেলেছি। গো লাইন বাই লাইন অফ দ্যাট টপিক, মেক ইউর ওন স্টাডি মেটেরিয়াল, মেক ইউর ওন নোটস, রাইট ডাউন ইন ইউর ওন হ্যান্ড, নিজে নিজে নোট বানিয়ে কোশ্চেন সলভ কর। টিল দ্য টাইম ইউ আর বিং এবল টু সলভ কোশ্চেন্স, then it is okay. Otherwise there is a problem in understanding. Clear? That's all about respiratory system in plants, respiration in plants. Fine, আজকে এটুকু থাকবে। So that's all about respiration. That's all about respiration. Yes, some of the questions are also there. Yes, end product. We have done this? No. Last day we have completed this. That's it. End product of citric acid and Krebs cycle. Chalo, do it. Yes. End product of citric acid Krebs cycle is citric acid or Krebs cycle is carbon dioxide and water. Carbon dioxide and water. Carbon dioxide and water. Out of 38 ATP molecules produced by glucose, 32 ATP molecules are formed from NADPS2 FADS2 in
C.
why EMP? EMP means only glycolysis. Why not oxidative decarboxylation? What is oxidative decarboxylation? It is not oxidative phosphorylation. What is Krebs cycle? Only Krebs cycle will be fetching you 24 ATP. Respiratory chain is going to talk about everything. Respiratory chain এর মধ্যে কি কি ঢুকে যাচ্ছে? Glycolysis, Krebs, link reaction and Krebs cycle. কি রে? বুঝেছিস সেটা? Great. Fine. Next. Tell me. Oxidative phosphorylation is production of. Oxidative phosphorylation is production of ATP in photosynthesis, NADPS2 in photosynthesis, ATP in respiration, and NADPS in respiration. ATP in respiration. Oxidative phosphorylation is production of ATP in, not in photosynthesis. Not in photosynthesis. Terminal cytochrome of respiratory chain which donates electrons to oxygen is cytochrome A3. Cytochrome A3. How many copper units they have got? Copper atoms. Complex 4. Complex 1 is having iron. Complex 2 is having iron. Complex 3 is having iron. But complex 4 is having 2 copper atoms. 2 copper atoms. So terminal oxidation এর সময় ইলেকট্রনটা কি অ্যাকসেপ্ট করে? A3. So this is the answer. আমার মনে হয় না আর কোনো কোশ্চেন্স আছে? Yes, that's all. Clear? Solve questions. Again I repeat every day. Solve 25 questions in physics, 25 questions in chemistry and 50 questions in biology. Do it on everyday basis and don't think about Neet. অত দূরে ভাবতে হবে না. Do this on a regular basis. You are going to crack Neet. No issues about that. Clear? So what we are going to do? We are going to do in the very next class, we are going to start with a new topic again, which is going to be a very important one. Clear? That's all about respiration plants. Go through the 4 topics that we have already covered. 4টে পার্ট বানিয়েছি আমরা respiration plants এর। পুরো ফার্স্ট টপিক থেকে নিয়ে 4th টপিক অবধি পর দেখে যা, কারণ কি মাথা রাখবি এইটা ভুলবি till the time you are not making your own notes, এই চ্যাপ্টারটা ভোলার চান্সেস খুব বেশি। This is such a chapter, if a teacher or faculty wants to make questions where you are not going to be able to make those answers, then this is the right chapter. Clear? So it's a tough topic. It's not hard not to crack it, but যদি একটু সময় দিস এটা পেরে যাবে। That's all about respiration in plants. ঠিক আছে? চলো।